everybody. I'm Brent. I'm a theoretical neuroscientist. I've recently moved to the University of Chicago to be a member of the Department of Statistics and the Department of Neurobiology. Previous to this, I spent a long time at the University of Pittsburgh in the math department there with Bart Ermitrout and John Rubin. I was there for my formative faculty years. Previous to that, I was at NYU, and I see Lai Sang Young, her video is open, so she was there when I was there, working as a postdoc with John Renzel. And then previous to that, I did my PhD work in Canada with André Longtin, who I think is on here as well, and Leonard Mailer, mostly Electric Fish. So happy to be here. Thanks so much, David and Carl, for organizing this. I think it's a great thing for our community.
So, thank you very much for your introduction.
No problem. Cool. Should I share my screen?
You can share. Yeah, sure.
All right. Can everybody see? You can see the screen? No problem. Okay. So this is a new talk. This is my first time I'm ever giving this particular presentation here. So if it's a little rough on the edges, I apologize, although I tried to polish it as much as I could. And I'm really looking forward to feedback from people here. So the way I think about this project is I work 6 days of the week at least. This is a funny project for me because on sort of Mondays and Wednesdays and Fridays, I think I'm working on something important. I think the result has something to say that hasn't been said and people should hear. That's what I feel on Mondays and Wednesdays and Fridays. But then on Tuesdays and Thursdays and Saturdays, I think this is an absolutely trivial result that is not difficult to understand. I hope I can convey that here. So maybe this is kind of the mark of, I guess, maybe a good result in theoretical neuroscience. I don't know, where your sort of result is at the boundary between saying something important, which I hope I am. I'm not sure. That's up to you guys to judge. And then it's relatively straightforward to understand. I don't claim to be doing high-level mathematics here. Sorry, Bard. I know Bard's on, but this is going to be relatively straightforward. I hope everybody can follow. Okay. I work in cortical variability. That's one of the things that my research group really focuses on. And this has been known for a very long time. This is one of the earliest recordings from Cortex. It's from Lee and Jasper. They're Canadians. They're at McGill. And they were one of the first people to really record individual units from Cortex. I believe this was from a cat. And the thing that's very striking, and I think has been said over and over and over the years, is that the discharge times of neurons are fairly erratic, right? There's this randomness to when neurons decide to emit an action potential here. This is perhaps a little bit more modern view of this. This is from a paper from Robagoras, Tony Movshin, and Eros Minicelli, where they were reanalyzing data from Tony's lab, I believe. And here they measured variability by saying, okay, we're going to count the number of spikes that a neuron produces, N, and we're going to look at something called the Fano factor, which is the ratio of the variance of the number of spikes in some window that they're observing the system divided by the expected number of spikes here. And across various levels in the visual hierarchy, we see that the variability is quite impressive here. A Poisson process would be exactly on that dash line here, where the variance is equal to the mean. And in most of Cortex, especially in deeper layers, deeper areas like MT, the variability can be much, much higher than you would expect for a Poisson process here. So this is a nice paper that I encourage you guys to look at. They have some interesting models, statistical models to discuss why this is the case. Why is the brain variable? Well, maybe one reason would be physiology is just sloppy. This is as best as we can get, right? Biology is just not great at controlling itself. And I don't think that's true. There's lots of examples in biology and physiology where variability is far less than we see in the cortex here. Here's a couple different examples. The cardiac, the heart, even though there's certainly interesting aspects of variability in the heart and people study it, it's not anywhere near as variable as a neuron in cortex. There are also sort of neurons in other areas of biology here. So this is sort of classic work from the somatic gastric ganglia and Crab. this is the chunk of neurons that are responsible for chewing, controlling the stomach of the crab for chewing. They're extremely rhythmic. This is my old love, the weakly electric fish. It's has a bundle of axons that produce an electric field around the animal, and it's one of the most regular oscillators, I think, in biology. The deviation, the cycle-to-cycle deviation here is like 10 to the minus 4. It's extremely reliable in terms of its periodicity. And then Zach Mainin and Terry Zignowski did some famous experiments over 20 years ago, where they took a neuron in isolation, so it was an in vitro preparation, and they would feed in a time varying signal over and over and over again for, in this case, 25 trials. And they saw that this neuron, when it was isolated from a bunch of other neurons in the cortex, can be quite reliable. So this neuron fired with the same spike pattern almost on every trial here. So I don't think the answer for why the brain or why the cortex is noisy is because the individual components can't figure out a way to be more reliable. I don't think that's the case here. So when we think about modeling the nervous system or the fluctuations in the nervous system, we sort of think about two different camps. Perhaps the variability that we see in a neuron is inherited from variability outside of the network, right? So this is really just an entrainment for a stochastic entrainment in the system. That's one framework. And then sort of on the other side of the spectrum here, we can have that variability as an emergent property. It's a reflection of the interactions within the system. So this, chaotic dynamics, I'm thinking more on the lines of turbulent dynamics that happens internally here. And lots of us have worked on thinking about variability in this way as well here. So there's sort of these two different frameworks here. And I'm going to compare and contrast these a little bit at the beginning of my talk here. So where do we sort of sit here, and how do we think about models that are emblematic of one of these two versions here? So this is sort of an outline of my talk. I'm going to review a lot of the data out there that has discussed how stimulus can quench neuronal variability, and talk about the network models that these data sets inspired. And then really sort of midway through, I'll talk about really new work, which is analysis of some voltage and current clamp in vivo data from a collaborator, Helena Desnik's lab. And this really disagrees with some of the network model predictions. And then we're going to propose a much simpler, and this is where I think, the Tuesday, Thursday, Saturday simplicity of the talk comes through, and how a much simpler explanation for some of these things has sort of been available. Okay. So I'll start here. So this is work from Mark Churchland and Byron Yu when they were in Krishnan Shenoy's lab. I think Mark was a postdoc and Byron was a PhD student at the time. And they have sort of a famous paper, now lots of us know it, where they looked at data. In this particular case, this is data from V1. I don't believe this is their data. They looked at this in motor cortex primarily here, but it's all, the story's the same. What they see is there's a stimulus that turns on around where the arrow is. In this particular case, it's some drifting grading here. If you look at the variability, the trial-to-trial variability before the stimulus turns on, so putatively in the spontaneous state, the firing rates of the neurons are low, but the Fano factor is higher than a Poisson process. So it's around, in this case, 1.3, maybe 1.4 here. And when the stimulus turns on, the firing rate goes up, and yet the normalized variability, the Fano factor, is decreased here. So this is what we mean by quenched. The variability seems quenched. They did a lot of controls here where they looked at subsets of their data so they could match the mean activity in spontaneous and evoked, and they still saw that variability is quenched here. So it's really that the trough-to-trough fluctuations in their data set are reduced during stimulation here. So this was one data set, and you'll notice sort of this paper has, I think, a ... here. I think this paper has something like 30 authors on it. They really did a tour de force to get data from a large number of labs, primarily working in, or I think all working in non-human primates, looking at different areas of the nervous system, and they saw the same story appear every time. So I just showed you the stuff from V1. I believe this is probably stuff from Marlene Cohen and John Monsell in V4. And then MT, 2 data sets, LIP. You certainly, even for in reward centers like orbitofrontal cortex, we tend to always have this quenching of variability as measured from the Fano factor that appears when that neuron is engaged in some task or some response. They did say, I'm not showing here, they also showed that variability is quenched when neurons are not engaged, so the stimulus is off-tuned for that cell, so the firing rates don't go up, the Fano factor nevertheless still decreases. So this is a pretty widespread, pretty reliable phenomena. This is in terms of variability controlling in the cortex, this is pretty rock solid here. So how do people think about this? So I think this is now a popular model where they say, well, spiking variability, we can decompose this into, there's some firing rate that is time dependent, and that firing rate is itself a stochastic process. So there's some stochastic firing rate, and then when you condition on that rate, then producing spikes is just a simple Poisson process. So we decompose variability into rate variability and spike emission variability. And a lot of people have done this. This is formally called the doubly stochastic process. So Teich, pushed this way back in the mid 80s, late 80s. And this is really, I think, the way that Mark Churchland and Eero Simoncelli and Tony Manchin and Rob Goris really want to think about these kinds of this variability. So here's the way I think about it, or they think about it. This, I think, is a review from Mark and Larry Abbott, where they say, well, this rate variability, the firing rate of the neuron is in orange here. And here they're showing, I think, 4 or 5 trials. And we see that on each trial, there's fluctuations in the actual rate of the neuron. And then, of course, they have to generate spikes given that rate. And when the stimulus turns on, of course, the rate goes up, so there's more spikes after the stimulation. But the trough-to-trough fluctuations in the firing rate itself, these are what are quenched, and all those sort of orange lines start to collapse around the trial average line, which is in blue here. And we've removed the source of variability. So the Fano factor in this simple framework, if everything's Poisson, would be 1. That's what you would get for a homogeneous Poisson process, plus whatever variability there is in the firing rate itself. And what stimulus does is it quenches this. This is really the idea for these explaining this data set. So that sort of philosophy or that modeling framework inspired several of us to go out and think about network mechanisms that could justify this view here. And here is sort of three different mechanisms. This is from actually a paper, the last one here. This is from a paper from Guillaume Hennequin, Mate Langel, Ken Miller, I think Yashar's on it as well, and Dan Rubin. But what they were doing is comparing and contrasting 3 different network frameworks. So one was from myself and Ashok Mukumar when he was a PhD student, and another one from Kanika, Rajan, Larry Abbott, and Haim Polinsky. So I'll compare and contrast these, but really this talk is in some sense about showing how there's an alternative, simpler framework. Okay, so I'll focus on first to talk about how Ashok and I sort of figured this out, and in terms of this axis of stochastic, so inheriting fluctuations from outside versus creating fluctuations inside. It really sits on this complex system side here. So this is Ashok. He was a PhD student in my lab. Now he's at Columbia as faculty member. This was one of his first real projects in my lab. And what Ashok did is he compared networks that are balanced excitatory, inhibitory style networks, compared them in sort of a vanilla Erdos-Renyi style connectivity versus a framework where you would cluster excitatory cells into groups here. So in this particular network, I think he chose 20 different clusters. And what that meant is that neurons within a cluster either connected with stronger weight or connected with higher likelihood in that cluster here. And then the inhibition was just unclustered. What you see is in the vanilla balanced network, it produces a roughly asynchronous dynamics. Neurons are variable here. But in the clustered framework, you get this metastable dynamic where individual clusters of neurons can exist in a high activity state, and they may transition into then a lower activity state, allowing other clusters to then be promoted into a high activity state and so forth. So there's this slow dynamic of firing rates that appears in these clustered networks. Certainly the way we should think about this is each individual cluster is some bistable potential, and the fact that you've got lots of them really means the system's metastable. And these switches between low activity states and high activity states, you can calculate these using sort of Arrhenius escape style formalisms here. And we can get that the system will, depending on the amount of clustering in the network, the cluster will either sort of exhibit in the high state or in the low state, and there'll be stochastic transitions exhibited between these. And then, so that's what we have. How does this go away? So that's really the source of the variability here. We have this rate variability you can see in the individual clusters. How does stimulation really affect this? So what stimulation does is we're only going to stimulate a subset of the clusters here. So in the unstimulated regime, all of the clusters are roughly equal. So there's a lot of symmetry in the system. So every cluster has the same likelihood of being in sort of the high activity state versus the low activity state. But when you stimulate, you only stimulate a few of them. Those ones transition so that the high activity well here becomes the more likely well. And because of the recurrent inhibition that's required to balance the system, then the unstimulated clusters here, they tend to exhibit low activity dynamics. And when you try this, so here's an example where we're only stimulating 5 clusters, I think out of our 20 here in this example. We're only stimulating these guys. We're stimulating them between sort of time point 1 to I think 1.3 here. And we see that before the stimulus, the Fano factor of all the neurons, this is the average Fano factor in our system, it's actually quite high. It's like 1.6, 1.7. And then when you stimulate the system, we're looking at the Fano factor of everybody here, both stimulated and unstimulated. But a neuron is, if it's stimulated, a neuron is very likely to be in the high activity state. If it's unstimulated, it's very likely to be in the low activity state on a trial by trial basis. And then the trial by trial variability collapses, we remove the stimulus, and then it sort of recovers back to where it was in spontaneous. So it's this sort of breaking of symmetry in the system that shifts the internally generated variability to be sort of less impressive in this evoked scenario. Okay. So then here's a comparison between sort of the clustered model and then Mark Churchland's data, and then the uniform model, the model without clusters. It doesn't, the Fano factor doesn't change whether you're stimulated or not here. The firing rates go up, but the variance goes up to match so that the Fano factor is invariant. Okay. So that was sort of that was that paper here. But now I'm going to go on the other side of the spectrum and then talk about a paper from Guillaume and Mate and Ken Miller, where they had a very different view of this. They were using a stochastic stabilized superlinear network. This is one of Ken Miller's models that he's really shown quite impressively that it captures lots and lots of data in the visual cortex here. And they wrote this paper to discuss this quenching of variability here. The way that their model works is it's a simpler model. There's no clustering. There's no interesting wiring. There's definitely a strong inhibition here in the system. So it's a recurrent excitatory inhibitory model. And importantly, they have a nonlinearity in their transfer function. So typically, Ken uses this threshold quadratic nonlinearity. It's relatively straightforward to use, and it's amenable to analysis. And what they saw when they took this model, and now they're taking noise from outside the network. So they're embedding this model in a noise bath, if you will, coming from outside. So this is this inheritance framework. If they, you can see up here, this is the network sort of without stimulus. They give it a low level of stimulus, might be consistent with spontaneous activity here. If you look at the voltage fluctuation, so here they're really looking at the membrane potential dynamics of a neuron in their model. You get high fluctuations here in the spontaneous state. And then when you stimulate the network, certainly the average voltage goes up, but the fluctuations around the average also gets quenched here. And this is it for just 2 values of input H. This bottom plot shows you it for all values of H. And this is sort of the takeaway here is that while the mean voltage goes up when you stimulate, as you might expect, the fluctuations in voltage, this is the standard deviation, it goes down in response here. And it's really this decrease in the input fluctuations that is this quenching of variability. So they're able to take their model and produce spike trains from their model using sort of a Poisson process style rolling of the dice. And they got a good match with experiments as well here. So that's an alternative network model. The way it works, here I'm going to actually steal some slides from a related paper that I did in my lab with Tatiana Kaneshiro and Gabe Ocker, really modeling work in visual attention from Marlene Cohen, but the mathematical spirit is very similar. So the neuron is the same basic framework. The neuron is nonlinear, so the transfer function of the neuron is nonlinear. And what's happening when you go from a spontaneous regime, so a low firing rate regime or a low input regime in green, and you go to the evoked regime, is you're shifting the point at which you would linearize the network. So here we're going to all do linear dynamics. The system is nonlinear, so the point at which you're linearizing has now shifted here in these networks. And when you put some noise in there, treat it as an Ornstein-Uhlenbeck process because you're linearizing it, and you can compute what the variance of the activity of the E cells are, for instance, in this particular case, we see that even though in the low state here, the firing rates are relatively low, when we shift in sort of the evoked regime here, the actual variance that you would report decreases. And the reason for that is the fixed point, because you're relinearizing the system. In the evoked regime, the fixed point is more stable, right? The eigenvalue, the real component of the eigenvalue here becomes more negative. So even though the fluctuations from outside the network haven't changed, their variance is the same, their structure is the same, the fact that you've shifted the operating point of the network to a regime where it's more stable, the system's always stable, but it's more stable in the evoked regime, then the system's better able to dampen these external fluctuations. And that's why you see the quenching of variability in these kinds of networks here. So relatively straightforward example here. And this is sort of what Ken and Guillaume and Mate were showing here, that this is sort of the zone of activity in the spontaneous regime here. And then this zone gets contracted here because the system's more stable and now fluctuations can't go around. I'm not going to I don't have time really here to talk about Konicka's work with Larry and Heim, but they talked about this in the context of in the spontaneous regime, the network was internally chaotic in a rate-based framework, which of course Heim has done tons and tons of work on. And then in this evoked framework, they gave a periodic signal, and that periodic signal suppresses the chaos. The effective Lyapunov exponent for this system becomes negative because you're now entraining this chaotic system to a time-dependent forcing, and it's that suppression of chaos with a time-varying input that explains this result here. Okay, so that's me sort of talking about that quenching of variability that was prominent in the data. Really allowed a bunch of us to, get paid, do some work, figuring out network models that could capture this here. But all these models have sort of the one thing in common here, in that they're working from this framework that the system's doubly stochastic effectively. We have some notion of rate variability, and then on top of that is Poisson noise. And if we look from the vantage point of here's some presynaptic population that's driving a representative neuron in the network. So we're going to pull out one of the neurons in the network, and we're going to ask, how does this neuron see this quenching of variability as we go from spontaneous to evoked? And in all cases, if we just write, what is the current that neuron would be getting? Well, here now it's getting K inputs from some presynaptic population. These are sort of delta functions. If we look at the variance of the current that it receives, and it's in this doubly stochastic framework, then we have sort of the variance we might expect just from the fact that the inputs are Poisson, but then we also have the variance coming in from the fact that the firing rates of these inputs are variable. And if we want the Fano factor to drop, then the reason for this is the variance in the firing rates within the network must drop. And that's going to be reflected in this neuron as the variance of its input currents are going down. So here, this single neuron in this network is really just slaved to the variability of the network itself. If that variability in the firing rates of the network goes down, the variability of its inputs will go down because it gets its inputs from the network. And then that's why its own variability will go down. That's a self-consistent answer here in these recurrent networks here. So that's sort of a prediction that we have here, that these models say that, well, if we look at the currents going into a neuron, when we go from spontaneous to evoked, we would expect that the trough-to-trough fluctuations of those currents go down. We already know the trough-to-trough fluctuations of the voltage goes down. I'll show that again in a second, but we need to check the variability of the currents here, okay? So that's where we're now going to shift gears into this second part here of the talk, which is this new analysis of voltage and current clamp data. So this talk really here is in large part done by Cheng Huang. She was a postdoc in my lab. Now she's faculty at Pitt. And we started analyzing data from Helena Desnik's lab at Berkeley. And this first set of data was collected by Julia Witt, who's now a professor in Freiburg. And they're doing, Julia was doing multi-unit recording of excitatory and inhibitory cells. So these were labeled cells. And we looked at this in layer 2, or they looked at it in layer 2, 3 to a drifting grading at various contrasts here. So that when the color is dark, the contrast is weak. When the color is light, the contrast is high. Here we see this is the firing rates of a bunch of E cells. These are primarily E cells I'm showing you here. Their firing rates go up with contrast in this transient framework. Their Fano factor goes down. This is, again, what we would expect. We saw lots of stuff from Mark Churchland in monkeys showing this. Well, it also happens in mice. And if we look at, the scatter plot, we see that in the evoked regime, the variance drops, even though the mean increases. And if we compute sort of the Fano factor, right during this period of time here, the Fano factor drops as a function of contrast. So the stronger the stimulus here, moving us further away from spontaneous, if you will, the more reliable, trough-to-trough reliable the network becomes. Okay. So now this is a different data set from the same lab. This is Hillel's. Hillel actually did his own recordings here when he was an assistant professor in his lab, and he did some whole cell recordings, same kind of stimulus, same layer 2-3 neuron, but now he's doing patch clamp recordings in vivo. And we see that absolutely the mean voltage of a neuron during high contrast versus low contrast stimulation definitely goes up. So there's like a 10 millivolt jump when the stimulus is strong compared to just a 2 millivolt or 3 millivolt jump when the stimulus is weak. And if we look at the trough-to-trough fluctuations, so Cheng analyzed this, the fluctuations for the high contrast regime go down here. And this is not normalized. This is just raw variance of the memory potential. The memory potential becomes less stochastic in the evoked regime here. Again, very consistent with what people have reported. David Furster and Nick Priebe have talked about this in the cat way back in almost 15 years ago. And this is true in mouse as well. And this is sort of the aggregate data here. Okay. But now that was in current clamp mode. What Hillel did is he wanted to look at the excitatory and inhibitory conductances in the neurons. So now he went into voltage clamp mode. So you can either clamp near the reverse potential of inhibition, so that when you do that, you can measure the conductance of the excitatory path, or the current of the excitatory path. And then the same thing is true for inhibition. And sort of unsurprisingly, we see here that when you go from spontaneous to, or low contrast to high contrast, we see a big increase in the mean conductance. So this is the trial averaged conductance for excitation. Same thing is going on for the trial averaged conductance for inhibition. They're really going up. But when you look at the trough-to-trough fluctuations of excitation and inhibition, they also go up here. So the mean goes up and the fluctuations go up. This is maybe what you would expect from a Poisson process, right? But where is the quenching of rate variability here? So the looks like the excitatory and the inhibitory currents themselves go up in fluctuations here. And this is sort of the aggregate data. This is the fluctuations in voltage go down when you drive the system hard, whereas the fluctuations in excitation and inhibition go up. And this sort of on the surface looks like it might disagree with this network prediction that neurons, the reason their output spike trains and their output voltage quenches its variability is because the inputs quench their variability. Well, this doesn't look like it might be the case here, okay? We have to be a little bit careful about this. It's a little premature to claim that, and I'll sort of argue this here. So if we just take a linear model so that the voltage is basically going to be, proportional to the sum of the excitatory and the inhibitory currents, and if we want to compute the variance of that voltage here, then, we can just do simple variance rule here. And then we have that the variance of the excitatory currents, the variance of the inhibitory currents, but we have to also consider the covariance between the excitation going into a neuron with the inhibition also going into that neuron here. and when you just take a simple sort of conductance model. But here we're not letting the input resistance of the neuron change. We get the following. And what I just showed you is the things in red, they went up with evoked, going from spontaneous to evoked. So the variance of the conductance in the excitation goes up, variance of the conductance for inhibition goes up. We can't measure this. You can't measure the covariance of the excitation and inhibition going into the same neuron. You can do it for two different neurons, right? But you can't clamp at the same reverse potential simultaneously. It's possible to do this in retina. Fred Riecke's done it by shifting quickly between the rest states, but it's really has something to do with retinal cells and how fast they are at adapting. In cortex, it's really not possible. So this could save the day, right? This could save our network models, that even though the red terms go up, the blue term will also go up, and then here, because there's a mixture of reverse potentials here, it'll actually be a source of negative correlations or negative variability in the system. And then that's why the voltage variability goes down, is because of this cancellation effect. So we can't test this with the whole cell data, but we can estimate this from the multi-unit recordings. So this is what we're going to do here. We're going to take this model that the conductance for either excitation or inhibition, alpha is just a placeholder for these. It's going to be the sum of the inputs coming from layer 2-3 to this neuron that's in 2-3. Here we're thinking of our postsynaptic cell as a 2-3 neuron. And then here's the inputs from layer 4. If we look at this, then if we want to compute the covariance of the excitation and inhibition into a neuron, we have to take all the 4 different combinations. So layer 2-3 excitation, layer 2-3 inhibition, layer 2-3 excitation, layer 4 inhibition, and so forth. And we have this measured from the multi-unit recordings that I showed you at the beginning from Julia Viet, not the whole cell recordings. And now we have to estimate what this effective strength of coupling and number of inputs into a neuron is. So what we're going to do is we're just going to let the proportion or the percentage of inputs, this really proportion of inputs that a neuron gets from layer 2-3 relative to the total inputs, that's what P is. We can measure the mean conductance from Hillel's whole cell recordings. We can measure the average number of inputs from Julia's extracellular recordings. We can measure the covariance of these as well. I'll show you that in a second. So we have a couple free parameters. What's the proportion of inputs, excitatory inputs, a neuron gets from 2-3, and what's the proportion of inhibitory inputs a neuron gets from 2-3? We can vary these here. So here's the data that Julia had. So this is the mean spike counts from the various 2-3 excitatory, inhibitory, or layer 4 excitatory, inhibitory guys. Their firing rates all go up with stimulus contrast, not surprising. The actual covariability of excitation and inhibition tends to only increase when we're talking about a layer 3 excitatory input, a layer 2-3 excitatory input compared to either inhibition from 2-3 or inhibition from 4. Stuff that's going on in layer 4, correlations aren't really being modulated much. This is pretty consistent with work done by Matt Smith and Adam Cohen as well. They've shown that laminar differences, and I believe also Valentine Dragoy's lab has done this as well, where people see that co-fluctuations between neurons tend to be more impressive and more malleable when those neurons reside in 2-3 than in layer 4. So this is consistent with this as well. So now if we take all of this data and then have a constrained model and we ask, okay, what would be the minimum of the ratio of the variability in the evoked state divided by the variability in the spontaneous state as we vary those 2 free parameters, number of percentage of excitatory inputs from 2-3, inhibitory inputs from 2-3, we see that if we want this data set to explain the fact that variability in the evoked regime is lower than the variability in the spontaneous, we have to live somewhere up here. So that this white line is the number, the 1 here, basically, when you're at 1. Anything above the white line agrees with the quenching of variability. Anything below the white line doesn't. And if we ask sort of where does cortex live, well, it probably lives at the star here. It's unreasonable to think that a neuron in layer 2-3 gets most of its inputs from 2-3, that's excitatory, and most of its inhibitory inputs from layer 4. That doesn't make sense. When people measure what these numbers really are, they're more in the star here. So from a data-driven point of view, we can sort of reject a current-based model for this as well here. Okay, so now that that sort of trivial part of the thing I think that is interesting but trivial of the talk, let's move to a conductance-based neuron model and see how well we can do. So why am I doing this? So let's go back to that whole cell data that Hillel recorded here. So what we can do is we can look at the autocorrelation function of the voltage, the membrane potential, during either spontaneous, that's in blue here, or when we go from low contrast to high contrast. We see that the membrane dynamics becomes quicker when you go into the highly evoked regime here. And when you fit this to an exponential, we see that the time constant of the membrane potential dynamics roughly halves as we go from spontaneous to evoked frameworks here. So this neuron is becoming sort of faster in its dynamics here. When we look at the same thing for the synaptic conductances into the neuron, that's not what happens. So the synaptic conduct, so the reason this neuron is getting faster in its dynamics isn't because the currents that it is getting are faster, right? The current, the excitatory conductance, the inhibitory conductance, they are not changing their time scales. These are well fit by a double exponential model here, but in both cases, sort of, you're not getting impressive changes in the time scale of these things. So I think people are starting to see where we're heading here. So, let's actually look at a conductance-based single neuron model. So let's go back to this single neuron here. It's going to get some bombardment of excitatory conductance. It's going to get a bombardment of inhibitory conductance. These are going to go through filters here. And this is now going to be a singly stochastic process. There's no doubly stochastic anything here. The spike trains coming down from this excitatory or inhibitory bundles here, they're just Poisson processes with a certain rate. That rate can change depending on whether we're in spontaneous or evoked, but we're not thinking of it as dynamically changing. Okay. So, it's hard to analyze this. So what we did is we do what most people do. We do a diffusion approximation. So this is just an example for the slide. We don't have to have this scaling here. But as the firing rates of neurons gets higher, and here, when I'm thinking of firing rate, really here, I'm thinking of the pooled rate coming down this chain, right? So this firing rate is the number of neurons here times the rate of an individual guy, okay? So when this rate gets into the 1,000 hertz, I don't think a neuron in the presynaptic pool is firing at 1,000. I think there's more like 100 neurons firing at 10 hertz, let's say, right? As we increase the firing rate of this presynaptic pool and we decrease the kick size associated with each one of these excitatory inhibitory hits, we sort of converge to sort of a smooth stochastic process here. Lots of people have done this. So I'm citing one of the early pioneers in this, Richard Stein, but many other people, Mandelbrot, Gerstein did it as well, all before I was born. So Thilo Schwalger spent a summer in my lab at Pittsburgh, and so we started talking about this observation we had with Chung. And so Thilo said, well, let's do a sort of diffusion limit from this. We're borrowing very heavily from work done by Magnus Richardson and Pierre Lansky and Lanska. But Thilo's done these calculations here. So here we have this framework where we go back to our conductance-based neuron. We have our diffusion limit for our excitatory inhibitory conductance. And here we're actually letting the fluctuations for E and I possibly be correlated here, right? So this is this Kronecker delta. So C here is the correlation between the fluctuations in excitation that a neuron receives and the fluctuations of inhibition that same neuron receives here, okay? So these guys did calculations for C is equal to 0. Thilo did it for arbitrary C. And this is what you can get. This is somewhat what you would expect. So we can compute the average conductance. We can compute the variance of the conductance. We can also compute the covariance between the conductance here as well. It's relatively straightforward. Then we can get these net passive features. So as you would expect here, the total conductance of the neuron is just sort of the sum, the resting voltage of the neuron in the absence of a spike threshold. Here I'm removing a spike threshold from this neuron. It's just a passive sac of potassium, if you will, at this point, or chloride. Here, this is the steady state, and then this is the effective time constant of that neuron. Remember, this is something that goes down for the neuron, and now we see that because here the average conductance will go up with the firing rates of that presynaptic pool, which you would expect would go up with the contrast of the stimulus, and then now the time constant will go down because of that, relatively straightforward. So indeed, when you compute, how does the mean voltage of this neuron compare as we sort of increase the firing rate of this presynaptic pool? So spontaneous would be somewhere here, evoked would be somewhat here. It just goes up. When we sort of try to understand this, it's relatively straightforward. When the firing rate goes up, there's this drive here. And here we've done it in a way that firing rate for excitation is double that of inhibition. So as we change this axis here, the excitatory cells driving this neuron, they increase, they have a doubling in the gain of their firing rates relative to inhibitory. So the overall mean potential increases because of that. We have this drive that goes up, but the fact that this isn't a linear line is you get this shunting effect here that causes this curvature here, because as you get closer and closer to the reverse potential here, you're gonna go down. Okay, so we have that for the mean memory potential. That's great, but then we can look at the fluctuations in the system too. This is a little bit more math, but we can get the variance of the voltage, which is what we're really after, and it's the sum of these three components, just like I showed you a few slides back when we were looking at the data here. And you can compute this for the stationary statistics framework here. And if we zoom in on sort of one of these terms here, we see it looks like this. And as we go through this same change in the firing rate, the fluctuations in the voltage actually go down. So this is a standard deviation of the voltage. It goes down, and it goes down again because of this shunting effect. And here we have a squared term here. And this square really interacts a lot with the time constant of excitation and inhibition, because your tau naught is 1/g here. But we get this shunting effect that really allows the fluctuations in voltage to decrease here. So when we compare these two things side by side, we see that in this simple model, we can have the mean voltage going up, and we can have the fluctuations in voltage going down. And this nice paper by Magnus Richardson really made this point that in these conductance-based frameworks, if you have Poisson inputs to a neuron, the mean and the variance of V can become untethered in this. So they don't have to necessarily follow each other. That's not true in a current-based framework. In a current-based framework, if the inputs are Poisson, if the mean input goes up, so too does if the mean voltage goes up, so too does the fluctuations. But in conductance-based frameworks, that's not true. So now we look at this data that we had motivating this, namely that the mean voltage goes up, but the fluctuations in the voltage goes down. This is what we're able to capture here. And the fluctuations in the currents are all going up here. So I'm not showing you that, but it's trivial in this case. The fluctuations in the currents are all going up. So this does a good job, at least heuristically, explaining this. When we now go back to this picture here, and we now let the input resistance of the neuron be state dependent, whether it's in the spontaneous or evoked. Now it's relatively straightforward to fit our model to this. So now this white line drops here. Here's when I'm taking this to be a factor of 2, because remember the time constant went down by 2. So that's the input conductance going up by 2 here. We can sort of get this regime to sort of exist where we would like here. Here now what we're doing is simulations of this for the full this is really a data-driven effect here. So rather than doing the asymptotics, the equilibrium statistics like Thilo did, we can just simulate this and fit this very well to Hillel's data. So what Chung has done here is we've used the time varying conductances, the mean conductances, as a way of constraining our model. And we can see that here, this simple model can capture not only the increase in mean potential and the decrease in the fluctuations of the potential we have, but also that the current fluctuations go up here. So this simple little model can explain the variability quenching that we see in Hillel's data, even though the fluctuations in the currents tend to go up here. So I only have a couple more slides, which is good. So I'm on time. So why is this important here? So stimulus evoked quenching of neuronal variability is very widespread, right? This is something that's pretty rock solid. There's lots of debates about noise correlations in the nervous system. I get in those debates for my normal day job. But no one's really debating that single neuron variability is quenched in the evoked framework. This is a great paper that took sort of a collection of data, and I think most people that have looked for this find it. So it's super widespread. Yeah, well, Ohm's law is pretty widespread too. right? This model of no one is this is not a controversial model. I'm not saying anything controversial here. This is the way we think neurons work. Certainly we can get more specific about this. We can have a spatially distributed neuron with dendrites, and that'd be fun to talk about. But from a point neuron point of view, this is a relatively straightforward model that no one has an issue with, and it's widespread, and this phenomenon is widespread. So that this parsimony is somewhat attractive here. The other thing I think that is important is when this model was put forward, this idea that we can take spike trains and think of their variability as dissected or decomposed into a firing rate variability and then some Poisson set of dice. That's a very convenient way to think of this data. And then all of these network models were proposed that fall into this class here. And this dropping of variability that we see that's so pervasive sometimes was thought of as a sort of a justification for this. But here, we don't have to have this doubly stochastic process to explain this quenching of variability in this simple conductance-based framework. If we did this with a doubly stochastic process, it would still work. So I'm not saying it's justifying a singly stochastic process model of the nervous system, but I don't need to invoke a doubly stochastic process now to explain the quenching of variability that is so widespread. So this puts a wrench in the justification for this view here. We need to find further justifications for this. Whereas I think previous to this explanation, perhaps we were very comfortable appealing to these network models to explain this, when allowing us to think about rate variability and Poisson variability as being different things. So to summarize, what I've really said here is that the increase in the total conductance can explain this quenching of variability in a conductance-based model, which is really just Ohm's law for second-order statistics, right? And this explains how the variance of the input current can increase, but the variance of the voltage decreases, which is, again, inconsistent with these doubly stochastic models. I'm thinking about people in my lab sort of trying to merge this with the network effects. I'm not suggesting the brain is not a network. Of course not. There's lots of interesting network questions in the nervous system. And now I want to start thinking about how variability quenching in conductance-based networks really happens here when you're sort of getting these things working. So this is the first time I've talked about this, to a public audience. A lot of this work was done, the analysis was done with Cheng Huang in her, dying fumes as a postdoc in my lab. The data is from Hillel Adesnik, Julia Veit, and then the conductance calculations were from Thilo. And this has really been done in collaboration with Ken Miller and Larry Abbott. So Ken and I and Larry have a grant, and sort of because I think we're all sort of vested in this interest, we've been chatting, and Larry and Ken have really helped sort of understand some of the consequences of this and really being skeptics for this model, which is good. So with that, I'll conclude. Thanks for your attention.